Okay, how's that there? Does it look okay? All right, thank you everyone. Thank you for the invitation to speak to you today. I wish that I could see you all in person, but I'm very happy to be able to tell you a little bit about the research that we've been doing on red squirrels remotely from afar. So as a network interested in phenotypic plasticity, it will not be news to all of you that the natural world varies in so many respects, abiotic ways, biotic ways, at all spatial and temporal scales. The recent interest in discussions at the Global Climate Conference about changes in temperature highlight the recent increases in global temperatures, but temperature fluctuates at all temporal scales as well, decadal scales, and smaller scales associated with El Nino or North Atlantic oscillation events, at annual scales, and even at diurnal scales. So on my bike ride in this morning, it was -7 degrees Celsius, and tomorrow we expect it to go up to 18 degrees Celsius. But some of these same variabilities in environmental conditions also happen in space as well, from landscape and biome scales down to the scales at which individuals interact with their environment, down to the micro scale that Louis Miguel was talking about in his introduction as well. Now, when we've got all of this variability in the environment, then optimal trait values are likely to vary from place to place and from time to time. And when these variations occur at large scales relative to the generation time and dispersal capacity of the organisms that experience them, we expect that those organisms will respond through adaptation. But when those changes in the environment happen at small scales, then we expect organisms to respond and cope through these environmental changes through phenotypic plasticity. Now, you're used to thinking a lot about phenotypic plasticity, but I thought I would just make reference to my definition of phenotypic plasticity as the capacity of a single genotype or organism to produce a variety of different phenotypes in response to changes in the environment. So here we have two pictures of different clones of trembling aspen and clones of Daphnia that each have been exposed to different environments. And the picture on the left then indicates what you might think of as passive phenotypic plasticity, where the different growth forms of these aspen clones occur because of different accessibilities to nutrients, sunlight, etc., that affects the form of these aspen trees. The example on the right with the Daphnia is quite a famous one because it's an active form of phenotypic plasticity. The two genetically identical clones were exposed to either the pheromones of an invertebrate predator, which resulted in the production of the large helmet and tail spine, or a genetically identical clone that did not experience these same chemical cues of a risky environment. In this case, it's an active and likely adaptive response of the Daphnia to increased predation risk. So in terms of plasticity and when plasticity is adaptive, I think of adaptive plasticity as occurring when the plastic response or the phenotypic response to environmental change that we observe is in the direction of the new fitness optimum. So as the fitness optimum moves to the left, if the trait distribution that I've tried to show in orange here also shifts to the left, then that would indicate adaptive plasticity. So when might we expect adaptive plasticity to occur in nature? Well, first of all, we need environmental changes that are large enough to affect natural selection, such that no single phenotype is best in all of the environments. Second, that these environmental changes are fine-grained relative to the generation time and dispersal capacity of the organism that experiences those environmental changes. And third, that the environmental changes are frequent enough to be relevant to selection at a larger scale. We might see evidence of very large environmental changes that have important implications for fitness. But if these are extremely rare on the timescale of the organisms that experience them, they're not likely to impose a strong selection pressure for phenotypic plasticity. And finally, the organisms need to have access to some sort of cue of these environmental changes if they are to adjust their phenotypes in anticipation of these changes in natural selection. And this is the way I think about adaptive plasticity, and it's shaped in part because of my experience studying North American red squirrels, which have all of those characteristics. They experience very large fluctuations in their environment that happens in a very repeatable way. And as I'll talk to you about today, they're able to anticipate those changes in their environment. So all of this work was done in the Yukon Territory, but I wanted to first remind you or point out that the red squirrels that you're used to seeing and experiencing in Europe are very different from the North American version of a red squirrel. And they're actually not that closely related. They're separated by about 13 million years of evolution. But they also differ in important aspects of their ecology. The Eurasian red squirrel is a fair bit bigger than our North American red squirrel. Our North American red squirrel defends exclusive territories from all other squirrels and is a conifer specialist. And instead of eating seeds and nuts where they're found in nature, the North American red squirrel hoards cones from conifer trees into a central place that they defend as part of their territory. So all of the work that I'll talk about today was done in the southwest corner of the Yukon Territory of Canada in a long-term study that we refer to as the Kluane Red Squirrel Project. And this work takes place within the traditional territories of the Champagne and Asiatic First Nations in the Yukon. And as many of you will be familiar with some of the studies that you have been running, this is a similar long-term study of individually marked animals that we monitor through regular live trapping events that was started by Stan Booten at the University of Alberta in the late 1980s. And now the project's led by 4 of us from 4 different universities who are working together to continue to study the red squirrels in this area. Over this time period, we've followed a lot of squirrels, performed a lot of mark recapture events, behavioral observations, and so on. And all of these things have allowed us to measure what we think of as interesting traits in the red squirrels and to appropriately track the annual survival and reproductive success of these individually marked squirrels. And although it won't play a big part in my talk today, by tracking relationships between mothers and their offspring and assigning paternity using microsatellite markers, we've generated a multi-generational pedigree that allows us to also do some quantitative genetic analyses of traits in this population. Now, one of the key features of the red squirrel system is that they live in an extremely variable environment. Their primary food source are the seeds of these white spruce cones. And white spruce is a masting species that doesn't produce consistent cone production each year, but instead goes through these episodic masting events. So the plot on the top is actual data from about 450 trees that are spread throughout our study area. And each fall we go and count the number of cones that are present on the tops of these trees. And it provides an index of the availability of food in the environment for the squirrels. And you can see that the availability of food fluctuates over 3 orders of magnitude from one year to the next. There are these extreme events called mast events every 4 to 7 years that I've just shown with the gray bars here, where the spruce trees produce a superabundance of cones, more cones than the squirrels could possibly consume or even cache. And that's a strategy that's evolved by the trees as a way of satiating this seed predator and enhancing the success of seed escape and subsequent germination. But following these years in which a superabundance of cones are produced, there are several years of very low cone production, and in some cases, 0 cone production, that is designed to suppress the population size of the seed predators so that the next time a mass event rolls around, there's fewer seed predators available to be able to harvest the cones. Now, the red squirrels buffer some of this variation in their environment by caching, by taking the cones and placing them in a central place that we call a midden. And this is just a photograph of quite a large midden. And all of the cone scales that are left over after the squirrel has eaten the seed pile up on the ground in this area. And there will be tunnels in these middens that the squirrel will use to store cones in the ground that keeps them fresh from opening and releasing their seeds, at which point they wouldn't be useful to the squirrels anymore. Now, another important aspect of the availability of food is when it occurs during the annual cycle. So cones mature and ripen on the tree in August, and they're cached by the squirrels in September. And that food resource then is used for survival through the winter and for reproduction the following spring. Now, most of the reproductive season within any one particular year is restricted to about a 1-month period, but it varies a lot from year to year. And so I've just indicated reproduction as ranging all the way from early February into June. So this is important because the reproduction by the squirrels is temporally disconnected from the availability of food. Okay. Secondarily, during these winter months, there's no fresh food available for the squirrels, and the squirrels do not hibernate, so they're active all winter long. And they must rely then on cached food in order to survive through the winter. And in many years, all of the reproduction that happens in the spring happens before the arrival of any fresh food in the system. So it's not until around May when the snow melts in this area and we get green up and the arrival of the new season's worth of food. Okay. So these masting cycles have really important consequences for the population ecology of squirrels. In mast years, when a superabundance of food is available, the population sizes of squirrels essentially doubles. And that's what I've indicated here by lambda, the population growth rate. And in non-mast years, in between the mast events, populations at best remain about stable, but they typically decline. Okay. And so as a result, we get this really strong relationship between the abundance of cones in the previous year and the population density the following spring. And so these pulses of food resources and the subsequent responses of populations in terms of the numerical responses have important consequences for natural selection. So the first question I wanted to talk about today then is how these resource pulses affect reproduction in red squirrels. The first thing I want to talk about is how red squirrels respond to food availability in terms of their reproduction. So here, what I'm plotting on the x-axis then is the abundance of cones that were produced in the fall as they influence reproduction the subsequent spring. So in springs following high cone years, we see red squirrels breeding much earlier in the season and raising faster growing offspring than in springs following years of low cone production. Okay. And so these are interesting responses, but I'm going to pivot pretty quickly away from these two traits and instead talk about the ways in which red squirrels anticipate food. So in addition to responding to food availability, red squirrels also adjust their reproduction in anticipation of the food that will be on the trees in August and September of the upcoming year. So we're talking about reproductive decisions happening in February, March, or April in anticipation of how many cones will be on the trees in August or September. And in this case, squirrels anticipate upcoming food mostly in terms of the number of offspring they produce. In terms of litter size, so litter sizes are larger in anticipation of large cone crops. There's an increased likelihood that yearlings will breed. And the most dramatic response is that in almost all circumstances, red squirrels will produce only a single successful litter in a season, except during mast years, in which they will recycle and produce a second litter within the same season. And all of these relate then to the total number of offspring that get produced within a year. So what I'd like to focus on mostly is this anticipatory response of litter size. So these data come from a paper led by Stan that was published back in 2006. But if we look at more recent data, including the past 28 years of data, the relationship is even stronger. So we have squirrels anticipating upcoming cone crops, and in years when it's going to be a bumper year for food, they're producing larger litters that are conceived back in February or March. So what you're probably wondering is how red squirrels are able to anticipate these upcoming cone crops. And the short answer to this is that we don't yet know, but we've got some decent ideas. It takes over a year to produce a spruce cone. The buds on the trees differentiate into either reproductive buds that will become cones or vegetative buds that will become branches in the summer before the cones are produced. And these buds are hormonally different. The vegetative buds have different hormones in them than the reproductive buds do. And we know that squirrels feed on these buds in the winter and spring, leading up to reproduction. And they seem to feed on the buds more frequently in advance of mast events. So we hypothesize that the squirrels are getting some sort of hormonal cue from feeding on the buds of the spruce trees that tells them whether those buds would become cones or branches in the upcoming year. And we suspect that this is the way that they know what the future cone crops are going to be, but this hypothesis has yet to be tested. Whoops. So the second question, though, is why squirrels increase litter size in advance of these large cone crops? Why increase offspring number and not some other aspect of reproduction in advance of mast cone crops? Well, natural selection on litter size is much like natural selection on clutch size in that there is an optimal litter size in red squirrels that results from a positive effect of litter size on fecundity, but a corresponding negative effect of litter size on per capita juvenile survival. And these two functions combine to result in optimal litter sizes that balance the benefits of increased fecundity with the costs of reduced per capita survival of offspring. And in most years, the optimal litter size in red squirrels is around the population mean of around 3 offspring per litter. But when we look at how selection on litter size changes during mast years, we can see, first of all, that mean fitness is much higher in a mast year than in non-mast years. But second of all, that during mast years, when there's a superabundance of food resources, and juveniles are able to create new middens and new territories of their own from all of this additional food that exists on the landscape, then the negative effect of litter size on juvenile survival is much weaker, if not completely absent during mast years, which results in a much higher optimal litter size during mast years than we see in non-mast years. So this suggests then that the anticipatory plastic response of litter size to upcoming mast events is adaptive in that it's in the direction of increased fitness in mast years. So what we've seen then is that squirrels increase litter sizes in anticipation of large cone crops, that this anticipatory plasticity in litter size is adaptive. And currently the cue that they use to predict upcoming cone crops remains unknown, but we hypothesize that it comes from hormonal cues on the buds of the developing cones on the trees. What's interesting is that in addition to these population responses to upcoming mass events, when we scale it down to the individual level, we find that not every individual is responding in the same way to these cues of masting. And so for this part of my talk, I'm going to pivot to some really nice work led by Dr. Lauren Petrullo, who's a postdoctoral researcher in Ben Dancer's group at the University of Michigan. And what Lauren did was to quantify how many of the squirrels responded to upcoming massed cone crops by increasing their litter size. And it turns out that only 53% of the individuals were responsible for these increases in mean litter size by increasing their individual litter sizes relative to what they would do during a non-massed year. So 53% of the squirrels are getting it right and anticipating these upcoming massed events and increasing their litter sizes accordingly. But we also see that about 27% of squirrels make a mistake and falsely read the environment and increase their litter size during non-massed years when these additional food resources are not going to be present in the environment. And what Lauren did was to apply what's called error management theory to this problem, where error management theory is simply about trying to understand how organisms ought to make decisions, given that information on the state of the environment is often imperfect. So we suspect that the squirrels are getting cues about the upcoming cone production that will occur on the trees, but these cues are not providing them with perfect information. So how should the squirrels respond or anticipate, given that this information is imperfect? Well, the basic idea of error management theory is that organisms should err on the side of making the less costly of the two possible mistakes. So in terms of responding by increasing litter sizes or not responding by keeping them small under these two environments, we have 4 different possible outcomes. One in which the squirrels increase their litter sizes and it is in fact a massed year, which is a true positive. One case in which they do not increase their litter sizes and it's a non-massed year, which is a true negative, and 2 ways in which the squirrels can make mistakes. Okay, increasing litter sizes when the food won't be there, and failing to increase litter sizes when it's in fact going to be a massed event. And error management theory suggests that we might still observe mismatches between phenotypes and the environment that might seem as though they're maladaptive in the short run, but these might in fact be adaptive in the long run because making one mistake is less costly than making another type of mistake. All right. And so what Lauren's calculations were able to reveal is that missing a mast and failing to increase your litter size during a mast event, okay, that's this false negative down here, is the more costly of the 2 mistakes. So there's asymmetries in the costs associated with making these 2 different kinds of mistakes. And so it's better to make a mistake and increase your litter size, it's not a mass, than it is to miss out on these really large fitness benefits that occur during a massed year. And not only that, but making more of these false positive errors actually makes you less likely of missing the mast and not increasing your litter size and reaping those large fitness benefits. So it suggests that there are squirrels that in fact have lower Q thresholds that are more likely to respond when they shouldn't, but as a result are in fact more likely to also respond when they should. And this leads to a much higher increase in fitness during these mast years. And so these asymmetries in the costs of responding to cues results in the persistent mismatch of false positives because the cost of making a false negative is in fact so large. So this is important because we often tend to think about adaptive plasticity through the lens of the benefits and the benefits of matching traits to a particular environment. But it's also important to consider the costs of mismatches and the potential asymmetries in the different kinds of mismatches that can occur. And that we might actually expect to see organisms that mismatch environments under some circumstances, and that this might be adaptive in the long run if these mismatches involve the avoidance of the more costly mistake in the alternate scenario. So in the case of the red squirrels, then, this suggests selection for lower Q thresholds so that they avoid missing out on the large fitness benefits of increasing litter sizes during mast years. So I've talked a lot about litter size, but I want to transition through the last part of my talk here to talk about the other component of this offspring size number trade-off, and that is the growth rate of pups in the nest. So we first access the pups in the nest by climbing up into the spruce trees and removing the pups soon after birth. And we do this again about 3 weeks later when they're old enough to permanently mark with ear tags. And the growth rate that I'm talking about here is the growth in body mass of pups from this time soon after birth until about 3 weeks of age. And as we would expect, females that raise larger litter sizes have pups that grow more slowly, and females that have smaller litter sizes have pups that grow more quickly. Now, natural selection on pup growth is not affected by the masting cycles directly, but instead is affected by them indirectly through an influence of population density. So in years when population density is higher, then pups that grow faster tend to survive better. And I've put a little asterisk on this statement down here because this relationship has changed through time. And this relationship is not as strong now as it was earlier on in the time series of the project. Okay, but the work I'm going to talk about today is from Ben Dancer's PhD work in my lab, at that time at Michigan State University, but Ben is now a faculty member at the University of Michigan. And Ben was interested in how maternal glucocorticoid hormones like cortisol might respond to population density and induce faster growing offspring in an adaptive way in response to cues of population density. So he was interested in the way that maternal hormones might integrate information from the environment and adaptively adjust offspring phenotypes to match that new selective environment. So in our populations, we follow individuals in a spatially explicit way. So we're able to quantify the density around a particular individual as the number of squirrels living within 150 meters of our focal individual. And that's what I mean by local density on the x-axis here. And Ben was able to use analysis of fecal samples to look at fecal cortisol metabolites in the feces from a very large number of individuals to find that females right around the time of parturition that are surrounded by more neighbors have higher levels of fecal cortisol metabolites than females that are in lower density areas of the grid. And this is a relationship that exists both across years as well as within years in response to spatial variation in population density. And now, importantly, these maternal fecal cortisol metabolites are positively associated with offspring growth rates. So females with higher levels of FCMs raise offspring that grow more quickly, which, if you recall, increases their survival under high density conditions. So this seems then as though there is an adaptive adjustment of fecal cortisol metabolites in response to population density. So this is the relationship that Ben observed from our natural observational data collection, but we also ran a long-term food supplementation experiment, which lasted for about 14 years, where in some populations, every individual in the population received a supplement of about 1 kilogram of peanut butter every 6 weeks through the winter. And this resulted in populations that were about twice the density of our control populations, which are shown in blue here. And in these experimental populations where we elevated population density through this experimental manipulation of food, in fact, females in these high density populations had higher levels of cortisol than females in control populations. But I think what was most elegant about Ben's work was that he identified what the cue was that females are using to assess population by performing a manipulation of the acoustic environment that females experience. So Ben experimentally increased perceived density without influencing actual density by taking experimental females and surrounding them by a set of speakers and MP3 players that played the territorial vocalizations of squirrels to trick focal females into thinking they lived in more high density environments than they actually did. And Ben, as a good experimentalist, also had appropriate controls where he played the calls of chickadees. And what he found is that females increased their cortisol levels in response to this perceived increase in population density above controls, both true controls that experience nothing and the chickadee controls that experience the sham manipulation of hearing chickadees around their territory. And so this suggests then that red squirrels are using vocal cues of hearing the territorial calls of their neighbors and integrating that information to be able to not only assess local population density, but to adjust offspring growth rates through these maternal glucocorticoid levels. So we have this typical trade-off then between offspring size and number, where females that produce larger litters produce slower growing offspring than females that produce smaller litters. But in these high density environments that are created through our food supplementation experiment, this trade-off is almost completely eliminated, where females are able to produce large litters of very fast growing offspring. And this is maybe not surprising because these females are supplemented with food. They've got plenty of resources. And so you might expect then that the additional resources of the food supplementation is what allows them to overcome this trade-off. But what's interesting is that when we don't provide them with any additional food, but we just trick them into thinking they have more neighbors, we see the almost identical elimination of this trade-off between offspring size and numbers, which suggests that females already have the capacity to raise much faster growing offspring than they do, but they only do that when they perceive the fitness benefits of producing faster growing offspring are sufficient to warrant the additional investment in those offspring. So here we have a situation where female red squirrels respond hormonally to vocal cues of local density in their environment. And these cues of increased density increase circulating levels of glucocorticoids in females, which increases the growth rate of their offspring. And these faster growing offspring then have enhanced survival when population density is high. So in this case, then, we've identified the plastic response being caused by the maternal plastic response in her hormones in response to a vocal cue of upcoming natural selection. So across these studies then, what I've tried to get across is that red squirrels plastically adjust their traits in response to cues of natural selection in their environment, and that these changes in traits are independent of the resources available to fuel those changes. So females are increasing litter sizes in anticipation of food being available later. And they're also listening to vocal cues and then increasing offspring growth rates, even in the absence of additional food resources available to fuel that. But I think the other important message from Lauren's work is that we might expect that traits don't always match the environment, but that these observed mismatches might in fact be less costly than alternative mismatches. And so mismatches might persist in nature, even though on the surface we might perceive them to be maladaptive. And with that, I'd like to thank the various funding agencies that have kept the project going over the past 32 years. I'd like to thank Agnes McDonald, whose trapline we work on, and the Champagne and Asiatic First Nations on whose traditional territory we work. This work was done in collaboration with the other principal investigators on the Red Squirrel Project, Stan Booten, Ben Dancer, Jeff Lane, and Murray Humphreys. And Dr. Lauren Petrullo's work was obviously central to the messages that I was talking about today. But today is also a day that we actually acknowledge as the Kluane Red Squirrel Project Field Worker Appreciation Day. And so I can't go without thanking the hundreds of field assistants that went into collecting the data over the past 30 or so years. And with that, I'd be happy to try and answer some of your questions and to engage in a bit more of discussion about plasticity in the red squirrels.
Okay, thank you, Andrew. So we don't have that many questions in the Q&A tab. Essentially, we have questions from Anne, but if you want to ask more questions, okay, so some are already being submitted. So I'll start with a question from Anne Charpentier. She's asking whether the cue could be from the birds. The cue from the birds could be olfactive as well as gustatory.
Yeah, I think we're going to let Anne ask the question if possible.
Oh, yes, yes.
So that we can see if she's happy with the answer. Just give me a second. Now I can. Okay, Anne?
It's better if so she has 3 questions.
Yeah, because she has several questions, perhaps they're related, etc. So, yeah. Anne, are you here?
Yes, I am. Can you hear me?
Yes.
Great. Hi, Andrew. Sorry, I didn't realize that I was the only one posting so many questions.
No problem.
But yeah, I mean,
you're not anymore, so it's good.
Okay, good. Well, actually, I don't know, because I have 3 questions and the first one wasn't the most important one, but I was just, you know, we have colleagues here exploring how birds could use olfactive signals that are emitted by bud trees. And I was wondering if you, because you said you're not entirely sure what the cues are, and you did mention that they taste the buds, but could there also be kind of a olfactive environment in years when there's a lot of buds around, then it creates kind of a smelly environment?
Yeah, it's a good question. And I guess we don't know. I don't know the answer to that. I just suspect that consumption is more likely, given that we know that they do consume a fair amount of buds during that spring season at around the right time. And at least superficially, it seems as though that consumption goes up in anticipation of a massed event. So through the winter, we do see more evidence of clipping and eating of the buds in advance of a mast year, which makes me think there might be some sort of feedback involved there. But you raise a really good point that there could be other mechanisms as well that we're just not thinking about.
Okay, thanks. Can I ask a second question?
Mm-hmm.
You can't see me, so that's a little bit frustrating. So actually, I guess just to go back to the first one, my thought was that if you have a strong smell, it gives you a better idea of quantity rather than the tasting, which perhaps doesn't give as much information about the production of the forest. And so that it's kind of linked to my second question, which is regarding the predictability of cone production. So I was wondering whether, I don't think you've mentioned like how predictable is cone production, how predictable are mast years, and also have you looked at whether this predictability is changing especially with climate change?
Yeah, so it's technically not a cycle in that like it doesn't meet the criteria of it being a cycle. So it's not that predictable. But on a time scale of roughly every 4 to 7 years, there is a mast event, although there was quite a long gap in between 1998 and 2005. And we think what happened is that in that intervening period, things were shaping up for a mast year, but pollination can get interrupted by weather events, which can then disrupt the cone production. And so so it's not predictable enough to be cyclical. And I guess in terms of changes in the regularity or predictability of the masting events, it's very hard to say, because even in the 30-year time series that we have, we still only have a very limited number of mast events. So anecdotally, in recent years, they've become much more regular in terms of the interval, but also the real absence of cone production in between those mast events. So the caricature would be a pulse of food followed by nothing, and then another pulse of food. But early on in the project, those mast years were interspersed with intervening years that sometimes had pretty good cone production. But the last 2 cycles that we've experienced have been really very dramatic, where the non-mast years have very low cone production. So, but it's just the last 2 cycles, so it's not very satisfying in terms of identifying trends. Do you have further thoughts on that, Anne?
No, I mean, I have another question related to the predictability, but I think perhaps there's other questions that should go before.
Yeah, I think we can go to a question from Guillaume Billet.
Yeah, do you hear me?
Yes.
Okay, hi. Thanks for the very interesting talk. I had a question, maybe, I don't know, maybe it's stupid because I'm not aware of the life expectancy of the squirrels, but is it known, or would it be possible to know whether those squirrel females, individuals that have increased the leaders on non-mast years, also generally have increased the leaders on mast years?
Yeah, thank you. So 2 important points. You know, given that time scale is important, I should have made reference to the longevity of the squirrels. So life expectancy in technical terms is less than 1 year, because so many of the juveniles do not recruit. But if they do recruit into the population, then they typically live 3 or 4 years, but they can live up to 9. So there's a long tail on that longevity distribution. But what this means is that there's a decent chance they experience both mast and non-mast years within their lifetime, but it's very rare for squirrels to experience more than 1 mast within their lifetime. So we've only had a couple of squirrels that have lived long enough to experience 2 masts. Okay, but your question was a really important one, and I didn't explain it well enough in when I was going through that part of the talk. But that is in fact what Lauren found. So the squirrels that make the mistake of increasing litter sizes during non-mast years are more likely to increase their litter sizes during mast years. And so the cost of that mismatch during non-mast years is more than offset by the benefit of hitting it in the mast years. So yeah, really good point.
Okay, we have several questions from Thomas Tully.
So maybe one of them, you showed us that the female are able to increase the growth rate of their offspring, whatever the food condition around. So which means that do you expect to have a cost to this kind of response, and are you able to detect such costs? I was maybe long-term survival costs or something like that, because otherwise you would expect them to grow the offspring as fast as they could every year. No?
Yeah, really good question. And you'll notice that in both situations, the squirrels are able to do more than they typically do. So they could raise larger litters, but they don't unless it's going to be a mast year. They could raise faster growing offspring, but they don't unless they have cues of high population density. And so, first of all, I think rather than this requiring some additional resources, the way I view the system is that squirrels are running well below their maximum in most circumstances, and they're only willing to invest more when there's a fitness payoff to warrant it. But fundamentally, there ought to be trade-offs that are present in some other aspect. But the squirrels are quite reluctant to reveal those sorts of trade-offs. They're quite frugal about compromising things like longevity, because longevity is very important to lifetime fitness. And so it's been very challenging to be able to identify long-term costs of these, short-term adjustments. So in some recent analyses, in previous analyses, we couldn't find very many of these at all. So we knew they had to be there, but they were very hard to detect. And so we suspected they were pretty small. But recently we've started to do some models of the growth rate, litter size trade-off and effects on adult survival that suggests that when they do push that trade-off really hard, it does compromise their survival. So there is some evidence of that, but man, it's hard to find. So it's not big.
Maybe we have time for you to ask another question, Thomas.
Okay, so I was wondering if you observe some individuals that completely skip some reproductive events, especially in the non-masting years.
Yeah, another really good question. And we do. And we have not investigated that much. That's something that we need to investigate more. So not every female breeds in every year. And we are now pretty good at detecting pregnancy by palpating the abdomens of the females. And so it's not simply a matter of them initiating reproduction and abandoning it. If they are doing that, it's at the very early stages before we're able to even detect fetuses. I think they are in fact skipping years, but we haven't explored that very much. So that's something to look into a lot more.
And maybe a last question, but
yes, you can.
Okay. I mean, we can imagine that the anticipation of the litter size is determined by the female, but I was wondering if you hypothesis that the male could also play a role. I don't know if they form a stable couples.
No, they don't. So there's very high rates of multiple paternity within litters. So females come into estrus for just one day in the spring and leave their territory, and they're pursued by say a dozen, 15 males, and she will mate with 8, 10, 12 males for a litter of 3 or 4. And rates of multiple paternity within the litters are very high. So there's no stability to or any pair bonds or anything like that. And then that's the only interaction that the male and female have is on that single day. So there's very little opportunity for males to contribute anything beyond the paternity. Although I guess I wouldn't rule out entirely your suggestion, but there's very little capacity for it. I will, maybe if I have a moment, briefly mention something to do with the males that is something we're actively pursuing right now. But another thing that happens during mast years that's quite interesting to us is that rates of litter loss go up quite dramatically. And we have some evidence of infanticide. And we think the reason litter loss is going up during mast years is because of infanticide events by the males. And females do all sorts of amazing things to try and hide their pups, oftentimes moving their pups hundreds of meters off their territory, hiding them in not even in squirrel habitat. In the most recent mast event, not a single female raised her pups within her territory. They all moved them off their territory, we suspect as a way of avoiding infanticide by males. And so I mention this because I think that both the females and the males are aware of the cues, whatever those cues might be, and it's affecting male behavior in addition to female behavior. And this is something that we collected a lot of data on in the mast year of 2019, and we're planning to collect the same intensive data on in 2020, but the pandemic hit, and so we had to kind of scale back to bare bones data collection. But we're interested in revisiting that question to try and understand if infanticide is the cause of the high rates of litter loss and why females are engaging in such fascinating behaviors to move their pups well away from their territories, but only during mast years. Sorry, that's not quite what you asked, but I kind of took it in a different direction.
Thank you.
Okay, we have one more question from Vaishnavi Purushottam.
Yeah, hi. So I was wondering if the presence of predators could also be associated with increased calls of the squirrels that live around, and would this, be potentially having an influence on their behaviors and the dynamics of the system that you are studying?
Yeah, really good question. I've really focused on sort of bottom-up processes here. The vocalizations that I was referring to are specifically territorial vocalizations. So they're used to advertise the owner's presence on their territory and to actively defend the territory. The squirrels have different vocalizations that they use in response to the presence of a predator, which might also influence things like glucocorticoid levels, but don't seem to be involved in this particular pattern that I reported here. As a bit of context, this long-term study of red squirrels is all done within the same ecosystem as the long-term studies of snowshoe hare and lynx that have been so influential in our understanding of predator-prey dynamics. And so we have a lot of data on the abundance of predators in the areas over this time period as well. But we don't often focus on that because in many respects, the top-down influences of predation on red squirrels seem to be, at least based on what we know so far, seem to be far less important than the bottom-up relationships between red squirrels and white spruce. And so the predators are there, they're eating squirrels, especially when the snowshoe hare are rare. The lynx switch to eating squirrels. So they are getting eaten. It is important, but it doesn't seem to be a strong selective pressure on the squirrels overall.
Right, thank you.
Okay.
Thank you. Maybe I should turn to the panelists, Luis Miguel, Céline, or Vincent. Do you have any questions?
Well, I would have several questions, but I have perhaps a comment or something about. I thought very interesting this thing where you showed that it may pay off to make some mistakes in some cases. And actually, I wonder if this can sort of be understood more broadly in the context of asymmetric fitness functions, because this what you described. So it's more like a comment, but you reminded me of a paper by Urban, was the first author, I think, a few years ago, where they showed that when you have asymmetric fitness function at the individual level, then when you're talking about fitness landscape of mean fitness against mean trait, then this can select for a trait that deviates from the optimum. And they actually use that for excessive immune response and autoimmune type of things. And it really looks like something a bit similar, but in the context with plasticity, where you have maladaptive misinformation, but still it's more costly to do the wrong decision. So
yeah, no, I agree. That I think you're right. I think that is a useful way to think of it. And as I might have mentioned, and in my sort of struggles to communicate Lauren's interesting results clearly, these are relatively new ideas that we need to think more about. So I appreciate that feedback. I think there's a lot of potential in this, but I think there's definitely more to do and more to think about. But I think the broader lessons, I wanted to mention it in part because it's new and I think it's exciting, but also I think the broader lessons are really important because we do tend to interpret from snapshots, right? Like we see organisms in their environments and we try and make inferences based on like a fairly limited window. And so I like the idea that we have to be a little bit careful when we do that, because situations that can appear maladaptive in that window might not be so in a broader context.
Yeah, and so another question that I think it's been sort of touched upon by several people, but I think it's about the individual life history and whether an individual that experienced a year where they have a very large litter size, is it followed in the next year by a lower investment, and can you detect these types of things? I think you sort of answered, but I'm not sure. Perhaps I wasn't paying enough attention. Do you see these types of effects?
Yeah, no, not very much. I mean, we have to really look hard into the data. So evidence of cost of reproduction is limited to the very young and the very old, for example. But otherwise, the prime-aged individuals do not show evidence of cost of reproduction. And so I think this just speaks to how careful the squirrels are in their investment. But they have to be there in principle.
Yeah, in principle. Otherwise, they will always have large litter size, right?
Right.
That's what I would think.
Yeah. I mean, the one window that we're starting to get now is we're now up to 9 years of data where we've now been quantifying the size of their caches. So now we measure how many cones each individual has stored in the ground. And I think that has some more potential to be able to reveal these costs. So we might not see the costs at the level of their life history, but the costs might be present in terms of the size of their stores. So it's important to remember that squirrels store almost all of their energy in the ground. Almost none of it is on their body. And so we don't get much in terms of insights from body size or body condition. They're not relevant. It's all about how many food resources they have stored in the ground. So it's possible that those metrics might reveal more in terms of costs.
Interesting. Thanks.
Céline, any questions?
Yeah, one or two, I think. One, I was wondering, with the data you showed at the beginning with masting, and like the last masting event, it was super, super high.
Yeah.
You also mentioned that it was kind of a strategy of saturating the predator. So do you think it can have an effect, or nothing detectable will happen?
Yeah, so we do have good evidence that it does in fact saturate the squirrels. And so we have measurements of the functional responses of the hoarding behavior of the squirrels to show that it gets saturated at high cone productions. Also that the numerical responses work. And for about 7 years, we measured the fitness of a whole bunch of trees within the population. And the seed escape, which we use as a measure of tree fitness, is one of the main determinants of that is the local density of squirrels around that tree. So when a squirrel lives near a tree, then it has lower fitness than when it's farther from the squirrel. So anything that the trees can do to suppress the abundance of squirrels in the area will enhance their fitness. So so that part of it is worked out pretty well. And it was 2019 was probably a very successful year for the trees because of the really large cone production. But also, I mentioned the squirrels doing these unusual things during mast years. And in 2019, they seemed so preoccupied with all of these off territory things, females moving pups off their territories, males chasing females around, that there was much less caching going on in fall of 2019 than we would typically see. And so seed escape should have been much higher in 2019. And so there should be a good cohort of recruitment of seedlings from that cohort. These unusual behaviors off territory seem to be increasing through time. We only see them in mast years, so we only have a certain number of snapshots, but it seems that they're becoming more prevalent as time is going on.
And the other thing I was wondering about is that you have a lot of variability of the cortisol for the same density level, or variability in how also they respond to cure of mast or not. And I was wondering if it was correlated with other traits, like for example, personality. Do you have like more aggressive individuals being maybe less stressed by density or these kind of things?
Yeah. Yeah. So now I'm not remembering. We do have relationships that we've tried to identify between personality traits and glucocorticoids. Sarah Westrick's work, but I can't remember. My memory is there's not strong relationships there. It's a little bit hard because there is a lot of variability. All of the observational data were within a 10-day window around the time of partition, because there is a lot of variability through the reproductive cycle. But there's also a lot of seasonal variability as well in glucocorticoid levels. And so some of those things will contribute to variability, although the data that I showed is just from that window around the time of parturition.
Thanks.
So I have one question, a very kind of detail-oriented question. You said that the most costly thing for the squirrels is to have what you call false negatives. That is to have a masting event and not produce more litter. But at the same time, just a slide before you said that 53% of individuals were responsible for the increase in litter size. So I guess it's not that easy to say that 47% of the squirrels were doing a false negative, but still I cannot make sense of those numbers. So if false negative is so costly to them, why only 53% are responsible? Is it just because of the differential reproduction between females, or?
Yeah, it could be. Well, I guess I don't know. I mean, because it should be higher. In the paper, we split out and that's part of why I didn't report the percentage of mistakes, because I reported the percentage of correct matches in the mast years, because there's sort of two separate ways in which you can make a mistake. You can fail to increase your litter size, and that would be just sort of like bad in a mast year. But then there's this complication of females producing two successful litters within a mast year. And so about a quarter of the females produce only one litter and maintain small litters. So that's the bad. But then about 25% don't produce larger litters, but they do produce a second litter. And so it's sort of a different way of producing more offspring during that year. But the same thing is true with the second litters. That is another way in which it is not a high proportion of females that produce two litters. And so the question is, why isn't that more frequent? Some of it has to do with timing. In order to produce two litters within the season, you need to start early in the spring. And so if your first litter is too late, then you don't have enough time. Some of it might be access to cues. Some of it might be having different thresholds for responses to the cues. But I think, your question is a really good one. It is surprising that there's not a higher percentage that are responding, because the fitness payoffs are really high. And so that's part of the question, is why? Like, what prevents the other females from responding?
Yeah, one reflex that we all have is that there might be a trade-off with survival to the next year, but then, as I understand, the year following a masting event is usually a very poor year. So I guess the trade-off here is not very a strong one. Yeah,
and I think mean fitness is like 4 times higher in a mast year than a non-mast year, and so all you really want to do is be around for the mast year, and then life is great, and what you do the next year is not really that important, because mean fitness following a mast is extremely low, because densities are really high. There's just very little space for juveniles to recruit, and so on. And so adult survival following a mast is pretty low. So the degree to which that reflects a wide-scale cost of reproduction, I'm not so sure, but it really is important for them to be alive and trying in a mast year, and then what you do after that is not as important.
Okay, thank you. Vincent, do you have any question?
Hi, no, I don't have any question. Actually, I wanted to ask the 53% question, but I've got my answer. No, I just wanted to say that it was really a great talk, and with super nice experimental data. So thank you very much.
Okay, thank you, merci.
And so, with this, I think we can really I'd like to warmly thank Andrew for this really stimulating talk, and also, Céline and Pierre for inviting him. So I'd like to just leave you two messages to finish. So our next webinar of this kind will be 25th of January with Tobias Euler, who will go a bit more on the developmental side of plasticity, I guess. And then the other message is that our network Plasticity, so for the people in France, they can register on the mailing list to that, and hopefully we'll go back to in-person events one day and meet and exchange. So with that, thanks everyone, and take care.
Thank you.